Hello class, today we are going to learn about diffusion. Diffusion is the movement of a substance from an area of high concentration to an area of low concentration. In this video, we will cover concepts such as facilitated diffusion, osmosis, and endocytosis. The membrane of a cell plays a huge role in the process of diffusion. The properties of the phospholipid bilayer allow for substances to diffuse into and out of the cell. For example, seeing that a phospholipid bilayer is made of phosphates and lipids, nonpolar substances like oil, fats, and other lipids can more easily diffuse. On the contrary, polar substances take more help to diffuse into and out of the cell. This is where facilitated diffusion comes into play. Similarly to normal diffusion, facilitated diffusion is the movement of a substance from high concentration to an area of low concentration. However, protein channels are needed to help move the substance across the membrane. These channels function as a doorway into the cell. Some protein channels require a stimulus, resulting in voltage-gated channels in which the electrical charge sends its signal to open the channel. Large molecules that need to move into or out of the cell require ATP and the assistance of vesicles and vacuoles. The following are all types of endocytosis. First, phagocytosis, or cellular eating, can be compared to Pac-Man. Penocytosis, or cellular drinking, is the movement of thick liquids across the membrane. Lastly, receptor-mediated endocytosis relies on a signal to enact the movement. Next, osmosis is very similar conceptually to diffusion. However, this is only specific to the transportation of water. The direction of this movement is dependent upon the total solute concentration of the substance. Thus, there are three kinds of solute variation. Hypertonic solutions have more solute and less water. On the other hand, hypotonic solutions have less solute and more water. Lastly, isotonic solutions have equal solute and equal water. These concepts all have important effects on the cell. First, an animal cell in a hypotonic solution will gain water, swell, and possibly burst. This is due to the higher amount of water outside the cell moving into the cell where there is a lower concentration. For example, a paramecium is more hypertonic than the pond water in which it resides. Therefore, water continually enters the cell. Over time, this has caused the paramecium to develop a specialized organelle, called a contractile vacuole, which pumps water out of the cell. Similarly, in a plant cell that is more hypertonic than its environment, it will take on water and its cell wall will become very stiff. This stiffness in regard to plant cells is known as turgidity. Contrarily, when an animal cell is in a hypertonic solution, the cell will lose water, shrivel, and possibly die. To combat this, saltwater organisms take up water and pump out salt because they are immensely more hypotonic than their surroundings. Similarly, plant cells go through plasmolysis or wilting when in a hypertonic solution. And we hope that's helpful.